Hi, everybody. Welcome back to another mini lesson of the respiratory system. So this topic is going to be pulmonary ventilation. So before we get into that, I'm going to put this slide at the beginning of all of my physiological mini lessons, just so you can kind of see what we're talking about. So in the very beginning of the physiological part of the textbook, it introduces 4 features of respiratory physiology. They include pulmonary ventilation, right? So let's get some right here. So pulmonary ventilation, which is what we're going to talk about in this video. And then we have what's called internal respiration, which is over here, which is basically just the gas exchange of the tissues. We have external respiration, which is just the gas exchange at the capillaries of the lungs. And then we have gas transport for both oxygen and carbon dioxide. So that's how I'm going to break up these physiology lectures. I'm going to do this one on pulmonary ventilation. I'm going to do one on the gas exchange, the internal external respiration, and I'm going to do one on gas transport. So those are the 4 kind of features of respiratory physiology. And then I'll have another video on control of how the body controls your breathing rates and things like that. So let's get into our pulmonary ventilation topic. All right. So as you're reading through this chapter, you are probably seeing some names of old dead guys, and they are more well known in the world of physics and chemistry, but we do have to talk about them when we are talking about respiratory physiology. The first individual that we're going to talk about his remembrance in the scientific annals of history is Boyle. So Boyle's Law is a property of gases and volumes and pressures. So the idea is, if we are going to kind of write this in an equation, is if you increase the volume of a container, what that will do is it will cause a decrease in the pressure, right? And then vice versa. It's an inverse relationship or indirect relationship. If you decrease the volume of a container, that is going to cause an increase in the pressure. Okay, so just basic, very basic. We'll start with the basics. So before we even apply this to the lungs, what does that look like? So let's start over here in this box. So gas is atoms that are in a density that's so low that they're far apart from each other, and so they're not a solid and they're not a liquid, right? You guys remember our 3 phases of matter: solid, liquid, gas. So in a gas, gases are just bouncing all over the place. They're bouncing all over the place in this room, in your room, in our atmosphere. So gases, oxygen, nitrogen, water vapor, carbon dioxide, all of these gases mixed up in our atmosphere, bouncing all over the place. And so when they bounce and hit against a container, that's what causes the pressure. So imagine like when you blow up a balloon, you're inserting a whole bunch of gas particles from your lungs into this balloon, so much so that it's increasing the pressure. You are pushing all these gas molecules on the inside of a balloon and you're inflating the balloon. Gas molecules are bouncing on the inside, gas molecules are bouncing on the outside, but you've increased the pressure in that by blowing up the balloon. So in this first box here, let's say this is our initial size of our container. Okay, so here's the initial size of our container. The little red arrow is just kind of showing you the randomness of these gas molecules bouncing on the inside of the container. And that's going to set up a particular type of pressure, okay, our set pressure. Then let's say you're going to take this container and make it smaller, keeping all the same amount of gas particles in that container. So we're not losing any gas, the numbers of particles, we're just making their container a lot smaller. So if you are one of these particles, you're going to be bouncing around a lot more frequently on the walls of your container. And that's going to have an impact on the pressure, right? Because pressure is the result of all of the molecules bouncing around on things. So if you decrease the volume right here, you're going to increase Nope, I did that backwards. Let's get the right one here. If you decrease the volume, you will increase the pressure. And so this is what we are seeing in that picture there. All right, so then let's go back. We'll go back to our set size, volume container. The molecules are bouncing around. They're giving you your kind of starting point pressure. Now, let's take that container and make it big. So we're increasing the volume, keeping the same number of gas molecules there. So if we're going to increase the volume, as if you are a molecule, it's going to take you a lot longer to bounce into those walls. And so the pressure, that measurement of how often those gas molecules are bouncing on the inside wall of that container, is going to go down, right? So if you increase the volume, you're going to decrease the pressure. Now, when you are using Boyles Law in application to physiology of the respiratory system, you always want to start with the volume changes because that's what's going to drive the pressure changes. You can have it going the other way. If you decrease pressure, you might increase volume, but it usually is the changing of the volume that's going to have an effect on the pressure, at least for the respiratory system and how that's involved. So the other pictures that I have on the screen, oops, let me erase all this stuff so it doesn't look too messy, is now we can apply that to our actual body. So when we are taking a look at our respiratory system, which is housed in our thoracic cavity, we have to take a look at some of those characteristics. So I'm just going to go to kind of a whiteboard page here. So let's say we have our lungs, right? So here's my trachea, and here is the diaphragm, right? So here's my diaphragm. That's my thoracic cavity, and that's my trachea to the outside world, right? So it is a respiratory tract. It's open to the environment. Now, the units of pressure that we're going to be talking about in the respiratory system is called millimeters of mercury. And out here in the environment at sea level, right, we'll just keep everything at sea level, is 760 millimeters of mercury. So that's just atmospheric pressure, or 1 AMU. We're going to go with 760 because that's a unit that your book likes to use. If we are not breathing, right, so either at the peak of an inhale or the bottom of your exhale, you are at equilibrium. Not the moving in or the blowing out, but just at the tops of your respiratory. So if you inhale, exhale, inhale, exhale, right? So at the peak and at the valley, the reason why you're not inhaling anymore or exhaling anymore is because you've reached equilibrium with the environment. So what's happening on these inhales and exhales to allow air to move in and out for us? Okay, well, we are going to, in this little kind of drawing here, we're going to focus on the diaphragm. There are some other accessory muscles, but the diaphragm is the major muscle of respiration. So it is a dome-shaped muscle. Let me see if I can stand up a little bit. So it's a dome-shaped muscle that sits like right here, way up under your ribs, right? So it's like this, and your lungs are kind of sitting here. And so when you inhale, that relaxed muscle contracts and it gets flatter. So inhalation is a contracting diaphragm. Exhalation is a relaxing diaphragm. So it's kind of like this. In, out, okay? So that is what we're going to use to change that volume, right? So that volume can affect the pressure. So let's say, we'll erase our diaphragm here, and let's say we're going to inhale. So we're gonna make it flatter. Right? So we just increased the volume. So we should see a corresponding decrease in pressure, right? Because we just identified that Boyles Law. So what do you think, what volume or what pressure do you think that contracting diaphragm is going to change the inside of your lungs to? Is it going to be a big number? Is it going to be a small number? What do you think? You can look in the book and see. It's actually not that big. When we inhale and we flatten that diaphragm, oops, we go to about 759 millimeters of mercury. 1 unit difference, but there's a difference. So if you are comfortable with the idea of diffusion, right, things going from an area of high concentration to low concentration, you should be comfortable with this idea of pressure gradients as well. So things like to go from an area of high concentration to an area of low concentration. So if you have 760 out here and 759 in here, then you will be drawing air into your lungs, right? So, and then I'm at equilibrium. Equilibrium. 759, air is coming in, pushing that back out. All right, so that is inhalation. When we contract our diaphragm, making it flat, increasing the volume, decreasing the pressure, and basically all what's happening when we inhale is we are trying to create an equilibrium with atmospheric pressure. All right, so now let's do the same thing on the exhale, right? So this was our inhale. Now let's take a look at exhale. So let me draw my lungs here. So we'll start with kind of my at equilibrium, 760 outside, 760 inside. So that would be maybe like right here. Inhale, yeah. So now we're going to exhale. So when we exhale, so we've just inhaled, our diaphragm is nice and flat. So then when we exhale, we've got to make that diaphragm relaxed and more rounded. So now we are going to relax our diaphragm. And what do you think that's going to be as far as changing that pressure? So we are going to decrease the volume, which will cause an increase in pressure. So we're sitting at 760 atmospheric pressure outside. How much do you think that's going to change on the inside? Well, it might be just like the inhalation pressure, is it goes to about 761. Just 1 unit difference, but that's all it takes. So where's our pressure gradient now? So we have a higher pressure inside of our lungs compared to the outside. So air is going to leave our lungs trying to create an equilibrium, right? So that's this part right here, trying to leave. That's an exhale. Or maybe I got it backwards. Inhale, exhale. Anyways, so you are changing the volume of your thoracic cavity, and that change in the volume changes the pressure. And with that pressure change, air is going to flow into your lungs during inhalation and out of your lungs during a exhalation. And this is Boyles Law, right? So Boyles Law applied to our thoracic cavity. So if we go back and take a look at our thoracic cavity, we can see some of these key features is the diaphragm, right? So we can see it as it's decreasing, or sorry, contracting. When it's contracting, it gets flat. That's increasing the volume, decreasing the pressure, so air flows in. Okay, so air flows in. When we are taking a look at a relaxed diaphragm, like right here, our volume decreases, the pressure increases, and so air flows out. Okay, so it is this repeating pattern of inhalation, exhalation, inhalation, exhalation. And because our rib cage is flexible, it has some joints, right? So we've got some movable joints, ribs and sternum. With some of our accessory muscles will elevate our rib cage in addition to the flattening of the diaphragm. And then on the exhale, they will decrease that volume as our diaphragm relaxes. Right? And then just lastly, real quick with this graph down here, it's just showing that inverse relationship is if you increase pressure, right? So if you're going up on this scale here, sorry, increasing volume, the pressure is going to go down. If you decrease volume, right, you're going to see that the pressure is going to be higher. Some people like graphical representations of things, so you can use that. All right, so let's take a look at kind of some pressures that we're actually monitoring. So what we just covered, this moving of air into and out of the lungs, is this pressure called the intrapulmonary pressure. Intra means inside the lungs, intrapulmonary. So if you remember, we went from 761 to 759. It was a difference of 1 unit of pressure. And so that's what they're showing here. So equilibrium, We are going to exhale when the pressure inside of the lungs is at 761. We're going to inhale here when the pressure is inside the lungs is 759. So this is our inhalation and exhalation cycle. And it kind of fluctuates 1 over and 1 under of that 0 kind of equilibrium pressure with the atmosphere. This next pressure is called the intrapleural pressure. Intra is in between the pleura. So again, you have to go back to the anatomy parts if you haven't covered those yet. But if you remember, we had our pleural membrane. We had our visceral is on the surface of the lungs, and the parietal is on the wall of the cavity. And there's fluid inside, that pleural fluid, that viscera. So whenever your thoracic cavity is pulled on, right? So if this is my thoracic cavity with the parietal pleura, it's getting pulled by the diaphragm, by your ribs increasing, kind of being pulled up. That pulling of the pleural membrane, that pleural parietal pleura, is going to, because there's fluid in there, it's going to pull on the visceral pleura, and it actually creates a negative pressure. So the pressure in between your visceral and parietal components of your pleural membrane is negative pressure. And that's really important because that's going to allow for your lung tissue to be increased, because we've just been talking about your thoracic cavity, right? So what if the cage is changing in size? How does that affect the lungs? Well, it affects the lungs because the pleura pull on each other. The parietal pleura, as your thoracic cavity is increasing in size, pulls on the visceral pleura because of that negative pressure. Okay. And then lastly, that's shown here on this graph is called the tidal volume. So tidal volume is the amount of air in milliliters that you inhale and exhale on just a basic quiet breath. So it averages around 500 mils. So 500 milliliters or half a liter is what we call our tidal volume. So inhale, exhale, inhale, exhale. So if I were to measure that, like a little measuring tool to capture all my air, every time you move air in and move air out of a normal quiet breath, this is what we call tidal volume. Okay, so here's just a picture showing some of those accessory muscles. So we had the diaphragm is our primary muscle for changing the size of your thoracic cavity, your inhalation and exhalation. It's your major muscle. It's working all the time. We have some accessory muscles for inhalation and exhalation. So some of our accessory muscles for inhalation, anything that's going to pull those ribs up, right? So scalenes. So if you remember, we learned about scalenes, pectoralis minor, serratus anterior, and external intercostals. All of these are going to be working to pull up and out on our thoracic cavity, increasing the volume, decreasing the pressure, causing air to go into our thoracic cavity. For exhalation, there's some muscles that we aren't familiar with and some that we are. So your rectus abdominis, internal intercostals, we've seen those before. Those kind of pull down on your rib cage. One that was not on our muscle list is called the transversus thoracis. Those help to pull down on your ribs closer to the sternum. So, and you can see those here, kind of in this anterior view, these little guys pulling down on the ribs. So between the combination of your diaphragm as your major muscle of inhalation and exhalation, but when we start getting into breathing patterns like forced breathing, like when you're exercising, you're going to pull in some of these accessory muscles to do some deep, faster breathing. So diaphragm's the major one, but know that there are some supplementary and accessory muscles for respiration here. Let me check my notes to make sure I'm not missing anything for this section. Okay, so just some terms here when we're talking about breathing patterns. The notes mentioned eupnea, E-U-P-N-E-A, is normal breathing. Apnea is less than normal rates of breath, and hyperpnea is higher than normal respiratory rates. We talked about that. Okay, so then we're going to talk about our respiratory volumes and capacities. So this is a lab you guys aren't going to get to do. I'm so bummed. I haven't figured out what I'm going to do for you with this yet. Hopefully I'll have it figured out by the time you're watching this video. But you should still be comfortable with these different types of volumes and capacities. So here is just a tracing. If you were to have a spirometer, you basically put this apparatus in your mouth and you just breathe with these different patterns, and it's able to measure the amount of airflow that's going into and out of your respiratory tract. So here is that tidal volume, right? Inhale, exhale, inhale, exhale. That's 500 mils. That's an average tidal volume. Now, let's say you're going to inhale, exhale. Your lungs still have air in them. They're not completely empty after just one quiet exhalation. So if you were to be asked to exhale as much as you can after your tidal exhalation, this is what we call the expiratory reserve. It would be like inhale, exhale. Trying to exhale as much as I can past, but just as much as you can. So when you take the difference between how far you can get and where you started, which was the end of your tidal volume, this is our expiratory reserve, which is on average around 1100 mils. For these are the male numbers over here that are on the chart, but you can also see the comparing female volumes as well. Okay, so that's our expiratory reserve. Now let's flip on the other side. Let's say you're doing a normal tidal volume, inhale, exhale, inhale, and you want to see how much more air you can breathe in over and above your normal tidal breath. Okay, so let's see. And that would get me here, and this is what we call the inspiratory reserve volume, the IRV. All right, so that is how much air you can go in over and above your normal tidal volume inhalation. So IRV and ERV, these are the abbreviations that are commonly used for these capacities, plus your tidal volume. So if you're to add your inspiratory reserve, your tidal volume, and your expiratory reserve, this is what we would call our vital capacity. Where's that? Right here. So it's from this point all the way down to this point, kind of this arrow that we have here. And you can also, you can add those values. You can measure your IRV, measure your tidal volume, measure your ERV, add those together. You can actually experimentally also measure your vital capacity. So breathe in as much as you can. Start measuring. And breathe out as much as you can, and then you'd have all that measured because you'd have the spirometer in your mouth, and that would be your vital capacity. Vital means living, alive. This is the amount of air that you can consciously control. Now, even when you do that, if you're blowing all the way out to what you can consciously exhale in the ERV or the vital capacity, your lungs still are not empty. There is a residual volume that's hanging out. That's a good thing because you don't want after every breath or a forced exhalation to empty your lungs of air because you'd stop all gas exchange. You'd probably slam close your alveoli, even with the surfactant, and then you'd have to take a lot of energy to try to open those alveoli back up again. So having that residual volume keeps your alveoli open, keeps gas exchange going, and makes the breathing a lot more efficient. Okay, so these are your respiratory volumes and capacities. You'll see something like this in the lab activity, the respiratory physiology lab activity that you don't get to do, but I will try to give you either data or some kind of representation of what the respiratory, the spirometry is what that's called. Okay, so we did that. All right, as I'm looking in the notes, I just wanted to talk one bit about this part right here. The respiratory rates and volumes and alveolar ventilation. I'll have to use this little bit of screen here. So in the notes, there is our respiratory rates. We have these variables, V sub E, or it's actually capital E. All right, so V sub E is your respiratory minute volume. How much gases, how much air are you moving within a minute? And so this is done with, that's not a good color at all. Let's do a different color. All right, so your respiratory minute volume is your breaths per minute, that's your F, times your tidal volume, right? So 500 mils. So if we were to average, say 15, let me get my calculator here, 15 breaths per minute times 500 mils per breath, that gives us about 7,500 mils per minute. Okay, so 7,500 mils per minute of respiratory minute volume is what that's called, which is very similar to cardiac output, right? Mils per minute. How much blood is your heart pumping? So how much air is your lungs moving? Are your lungs moving in and out per minute? It's about 7,500 if you had a respiratory rate of about 15 and your tidal volume of about 500 mils. Now then the next equation is called alveolar ventilation, this one right here, right? So moving 7,500 mils of air into and out of your lungs is great, but is all of that air going to reach your exchange surfaces? No, because remember we had a whole lot of conducting zone. So all of the air that's in your upper respiratory tract, your trachea, your primary, secondary, tertiary bronchi, small bronchioles, terminal bronchioles, none of that air is reaching your alveoli. It goes in as far as your lungs can take it in, and only a little bit of that air is actually making it to your exchange surfaces. And that's the really important number that will allow us to tell if somebody is actually getting air to their systems, to their lung tissue. So let's take a look at that one. This is called V sub A, which is your alveolar ventilation, which is we starting off the same with your breaths per minute, but it takes our tidal volume and subtracting what's called dead air space. By subtracting that dead air space. And so that would be all of the volume of your conducting zone. And it's going to be different from person to person, but it averages about 150 mils. So if we have a normal tidal volume of, say, 500 minus your dead air space, then that gives us this actual volume that's going to make it to the lungs of about 350. Multiply that by respiratory rate, we'll say 15, right? Let me get my fancy schmancy calculator here. So 15 breaths per minute times 350 mils actually making it to the alveoli. That comes down to a more reasonable 5250 for your alveolar ventilation rate. So this is mils per minute, which maybe coincidentally or not, is very similar to your average cardiac output. Remember, it's between 5 and 6 liters per minute. We are doing about 5.2, 5.25 liters of air per minute at an average respiratory rate of 15. So it's kind of cool that those two numbers are lining up like that. Okie dokie, so that finishes up this little mini lecture on respiratory, pulmonary ventilation, Boyles Law, respiratory volumes and capacities, and just a little bit of math of the alveolar ventilation rate and your, respiratory minute volume and your alveolar ventilation volumes. All right, so I'll see you in the next video with some more respiratory physiology. Bye.